Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about recombination of linked versus unlinked genes. To discuss this topic, we need to take a closer look at a non-sex linked Morgan cross. In this cross, we will not be using X or Y. These crosses are going to use autosomes. So the wild type fly is gray body and normal wing. It has two genes that we're looking at. We're going to cross that one with a double mutant. It has a black body and vestigial wing. When we cross those two individuals, we're going to get a heterozygous. So I've changed the letters to just regular A's and B's. So if we have a homozygous dominant for both genes crossed with a homozygous recessive for both genes, we will get a heterozygous individual for both genes. We've seen this before. Whenever you have a heterozygous crossed with a homozygous recessive for both genes, that is referred to as a test cross. So we will perform a test cross, and the outcome is 83% parental type and 17% recombination type. So if we look at the data, 965 resemble one of the parents, 944 resembles the other parent, the original parent. That means that we have 83% of the offspring looks like one of the parental types. We have 206 gray vestigial and 185 black normal. These two types do not look like either of the parents, so they are called recombinant offsprings. So 17% resembles recombinant offsprings. The data from this test cross suggests that genes are located on the same chromosome. How do we know? I've created a decoder key to help us understand how to identify when genes are linked versus unlinked. When genes are linked, the parental types will be greater than 50%, and the recombined types will be under 50%. So the previous example, we had 83% of the offspring looked like the parental type. So therefore, it should be linked genes. Now, if the parental types were 50%, exactly 50%, then the genes would be unlinked. So this is our decoder key that we can use to make predictions whenever we look at the results from a test cross. Since the data from the Morgan cross yielded a 83% parental type match, then the genes must be linked on the same chromosome. Morgan additionally proposed that some process must occasionally break the physical connection between genes on the same chromosome in order to get incomplete linkage. That physical connection was crossing over. Crossing over occurs between these homologous chromosomes. So that is what the physical connection was. So therefore, based on my decoder key, we're going to follow the red. Recombination of linked genes must have occurred. And to get the recombined types, crossing over must have taken place. If the genes were unlinked, the parental type offspring lookalikes would have been exactly 50%. We didn't have that in the previous cross. So there are two types of unlinked genes. The first one, the unlinked genes can be on separate chromosomes. Another example is the unlinked genes could be on the same chromosome, but they're so far apart that they act as if they're not linked, or they act like they're on separate chromosomes. So this is to help us understand what is going on. This is a homozygous dominant for both genes. When the chromosomes are amplified during interphase or replicated, then the chromosomes are going to go through meiosis 1 and meiosis 2. And eventually we're going to have 4 haploid cells that has one big A and one big B in each cell. These cells are haploid. This is the same process for the homozygous recessive for both genes. You're also going to go through meiosis 1 and 2 and produce 4 haploid cells, but these cells are going to have a little a and a little b in it. When you cross those two individuals, you will get a heterozygous individual. That heterozygous will have either a big A, a little b, or a big A with a big B, or a little a with a little b, or a little a with a big B. Two of those gametes resembles the original parent, and two will not. Now we have the sperm is going to sneak in. The sperm is also a haploid entity. It has also gone through meiosis 1 and 2. This sperm only has a little a and a little b because this is the test cross. Remember, the test cross is between a heterozygous individual and a homozygous recessive for both genes. You can also perform a Punnett square to see the results. So here's the Punnett square setup. Heterozygous for both genes crossed with a homozygous recessive for both genes. When you perform this cross, Punnett square, this should be your results. So unlinked genes exhibit independent assortment and random segregation. Therefore, the results from a test cross will be 1 to 1 to 1 to 1, or 25%, 25%, 25%, 25%. This would be the results from that test cross. The two cells in the middle are going to grow to look just like the parental types. The two on the outskirts are going to grow to look nothing like the parents. They're going to be recombined. So as you can see, the ratio is 25, 25, 25, 25. Unlinked genes exhibiting independent assortment only will yield a 1-to-1 ratio or a 50% to 50% ratio. This again is going to follow our decoder key, but to the red this time. This is if the parental types are exactly 50%, we know that the genes are unlinked. So this shows us the 50% that looks like the parental type. They are highlighted in red. Each unlinked gene moves independently of one another because chromosomes assort independently. That was the theory that Sutton and Boveri created. They created the chromosomal theory of inheritance in 1902 from observing the results of the Morgan cross. This theory states that chromosomes undergo segregation and independent assortment, not the Mendelian gene that is at a specific location.